Hello and assalamu alaikum from Pakistan Agriculture Research Center of Pakistan. Okay, thank you so much for inviting me. And let's have a look on AI in microbiology, right? So here we just have a research paper here, which is just showing that with the help of old identification method, the scientists are just trying to see the antibiotics, and they discovered the new antibiotics with the help of new deep learning methods to you can say fight against different types of infectious diseases in the human being, right? So with the help of machine learning, deep learning, AI, you can develop novel antibiotics to kill dangerous bacterium in the lab, right? So I am Dr. Shakira Ghazanfar, and I'm working as a senior scientific officer at Pakistan Agriculture Research Center. So here's some brief introduction of our lab ongoing project. We are just using deep learning, machine learning, and AI algorithms in our research to just filter out the different types of microbial strains to make a new product in our country, right? So we are also providing different types of microbial strains for research in our society as well. So there are some brief introduction regarding our team talking in different events regarding AI in microbiology research. So what is AI? Specifically, it's a, you can say, branch of computer science that aims to just create an artificial machine to serve a unique machine for the solution of different types of problem in my case, microbiology labs, right? So there is a domain like AI, machine learning, and deep learning. There's a difference between machine learning, deep learning, and AI. So you have to be very careful regarding selection of any AI technology to sort out any type of problem in your lab. So before going to start AI, you have to learn these basic terminology to sort out your problems. So here is the role of artificial intelligence or deep learning, machine learning, microbiology. In our, you can say, lab problems, or you can say microbiology, the different types of bacterial or yeast strains can be used in food as well as in antibiotics and medicine. But these bacterial, which having some type of potential, cannot be identified with the help of our own, you can say, old methods. So discovery and other type of validation process of any type of bacteria required extensive time as well as effort and budget. Therefore, it is great interest to develop a unique, you can say, screening method based upon AI to identify specific type of bacteria, right? Or it's a safe time, you can say, or and low error, you can say, technology to just screen out a bacteria from a huge type of bacteria strains. So if we are talking about, you can say, live microbial feed supplements, or you can say probiotics, these are unique probiotic strains which are naturally present inside human gut to improve the human health in cost effective and no, you can say, side effect. They can be used to treat cancer as well. They can improve the gut health. They can beat the, you can say, fight against bad bacteria as well. So these are naturally probiotics, but the selection of the probiotics is very difficult with old method. So, what are smart probiotic strains? These strains can be resistant to harsh gut environment, and they have a, you can say, huge potential to just, you can say, have a adhesion property to just hold a gastrointestinal tract of a host, or you can say human gut. So they have no various factor, and they generally recognized as a safe status from WHO as well. So there is a large of challenge in searching of smart probiotic identification. Major is to just, you can say, complex microbial genomic data. So if we are talking about the old identification method of the bacterial strain, which can be used to make a food product, they can be phenotypically identification problems, molecular identification problem, probiotic activity screening is another hurdle for the screening of smart probiotics. And then you have to follow huge test to just screen out a small tiny bacteria in to the lab for preparation of product. So it's a very, you can say, time consuming method. So different types of, you can say, challenges are available to just screen out as T bacteria or yeast strains in your lab for preparation of any type of, you can say, edible product. So if you have just say that why it's hard to prepare a target specific probiotic product in the market. Why? Number one major issue is that the target-based probiotic, or you can say, if you are setting a small yeast-based probiotics in your lab, you have to screen out the phenotypical test. Then you have to see the if you this screening test from a breed, feed type, environment, animal genomics. It's from, if it's from animal gut. So by using all these, you can say, parameters, you have to just screen out a specific bacterial strain inside the human gut environment as well. So if you are talking about probiotic product preparation without AI, you have to follow these 13 steps for commercialization of any type of one bacterial strain made probiotics. So you have to follow different types of WHO and FAO guideline steps. But if you follow the probiotic product with, you can say, AI, then it's very, you can say, time will be less for the screening of a probiotic product. So by using the AI method, you can screen out a unique, you can say, your own target bacteria from a huge number of strains in your lab. So how machine learning can help to search out target-based probiotic strain? They can easily speed up the probiotic, you can say, discovery. They can enhance the prediction accuracy, then can integrate the genomic potential as well, right? So here is a experimental proof of that, you can say, unique idea of using AI in microbiology. You have to see, in our lab, we have just developed a unique NGAB probiotic product tool, which can easily use to predict a unique yeast strains from a number of strains to build a new product in our lab. So we got a copyright from GPO as well. So what is the basic material method of that technology? Number one, we have just follow a unique, simple machine learning method to just screen out a unique probiotic strains from a bundle of probiotic, non-probiotics yeast strains, right? We have follow step 1 to step 7. These are all already available steps. We have just put our data, that is probiotic and non-probiotic strains. We have just follow the main, you can say, data collection steps in our lab, and then use this data for preparation of a unique model for screening of probiotic strains from a huge number of you can say, tested strains data. And for this, we have just gathered the data from WGS, whole genome sequencing from NCBI. It's a one data set where we have just screened out the data from our labs as well. We just use the already published data as well as the data from our lab as well. Then after using that, we have just, you can say, screen out a feature from this data, whether this have some, you can say, unique probiotic product related gene as well or not. Then after that, we have just went for, as you can say, raw read of probiotic bacteria, and after quality checking with different genomically available, you can say, tools, trimming tools, assemble tool, annotation, extraction probiotic, we just made our data in that condition. We can build a new probiotic, you can say, prediction model in our lab. Then in probiotic selection, we have just select our desired feature, right? Feature, gene expression level, presence of specific gene, as well as presence of a metabolic pathway as well. And then after that, evaluation relationship between different strain as well. Then environmental factor, conditions under different types of, you can say, selection. And then after that, the dependent of probiotic prediction as well. The ability of strain to confirm host beneficial is another feature in our data as well. Then after feature extraction, we just use a basic statistical test to just build on feature selection of our data as well. After feature selection and feature extraction, we just split our data into, you can say, test and train data into 20%. After using different types of model like GLM, RF, SVL, and NN, we just built a, you can say, our own desired model by checking the accuracy of these applied model. and then after evaluation, we have a just, you can say, give a validation of our data as well. Right after model, you can say validation, we have just different types of result in our hands as well. This is the data set we used in our lab to just predict a new yeast from a huge data set, right? This data can be, you can say, taken from the already published data available on the NCBI, as well as you can use this data from your own lab as well if you have some, you can say, mini objective in your mind, right? Then after that, EDA, exploratory data analysis, was done with the help of different types of, you can say, tools to just build, you can say, your own prediction, or you can say positive correlation between the colony morphology, colony prediction of your desired feature, given feature, right? So this slide show the feature important analysis, whether your given model having any type of accuracy, and our you can say model giving you can say huge prediction in term of, you can say, material accident. So the main you can say model is to just predict the accuracy of a pathogenic strain, the drug resistant strains, pH and acid tolerance strains. So overall, model prediction was very much you can say helpful for prediction of new probiotic strains prediction in our lab. So by using the machine learning algorithm, simple machine learning method, we just built a new model which can predict the probiotic and non-probiotic strains in our lab. By using this strain, we can easily predict the probiotic nature of that strain without going to perform any type of better test in the lab, right? So this is a summary of our AI-based prediction tool, data retrieved from data retrieved to have two model built up for the screening of unique strains from a huge untested strains. So it's the first page of our, you can say, NIGA probiotic predict model, where we just put our data, untested data, and can predict that, well, yes, this data can be used to prepare any type of unique, promising, or you can say novel probiotic product in future or not. So here's a unique, you can say, overview of our tool. Another overview of our tool, we just built a tool with the Council University Islamabad, and here we just built a new machine learning based tool for prediction of anti-cancerous peptides as well. So what is the main advantage of our NIGAB probiotic predict machine learning based tool? We can easily, we use this tool to screen out a unique strain, and that this strain is no use in our lab in our country to predict a new yeast strain for preparation of our new yeast extract production for bacteriatum, or you can say fluidatum, right? So another another ongoing project of our lab is Bacteriologene 102, AI-based Pakistani probiotic product for animal milk production enhancement. So all based upon, you can say, AI model to just build a new tool and then build a new product as well. So it's up to you. It's up to you. It's up to your lab. What type of problem in your lab? Use the AI tool and then build a new product if you have a AI expert in your hand, right? So after, what is the outcome of your machine learning tool? It can predict the prediction of draft genome as well, the pH prediction and draft genome, and easy to use, and it's a very user friendly and time safe for production product preparation as well, right? So after using the huge machine learning AI model in our research, we have a, as you can say, more than 300 impact factor from our lab, and there is our lab is just going to up with the help of AI algorithm in the research, right? We have a huge number of PhD MPhil students which are using AI model to build a new tool and build a new product. So what is outcome here is a unique, you can say, paper from our lab, which is just use AI model to build a new tool, and then it is publishing for computational biology and chemistry in 2024. Another paper which just use AI tool to be to predict the heat shock protein using machine learning model. So we are also using our expertise to just give a huge publication to the reader that the some book are under, you can say, writing process with the Springer Nature as well as CSE Press, where we are just using the concept of AI in probiotic selection to build a new model, right? So this is our another area. We have a research collaboration with the European Union, China, and the UK, where we are just using the concept of chat board for livestock. We are just trying to emerge, as you can say, and train the staff for AI working as a team member to build a new milk production enhancement, you can say, potato creation. So it's only depends upon your own, you can say, vision. What is your problem in your lab? As a scientist, you have to see the basic, you can say, learning of the problem. If problem in your hand, you can easily, you can solve sort out your problem, and you if this problem is sorted out, you can easily build a new innovation in your lab. If it's from microbiology lab, you can build a new product with the help of AI, machine learning, deep learning model. So let's learn AI and give a human as a safe environment for living. Thank you so much from Dr. Shakira, Pakistan Agriculture Research Center, Islamabad, Pakistan.